Welcome on my second set of notes. This is on Meiosis 1. So we'll just do the first part of meiosis. All right, I got a little picture here of meiosis and meiosis 1, and you can kind of see that it looks a lot like mitosis. Probably the only thing that's really different is, and if you would really pay attention when we get to the metaphase, that you have two sets of X's instead of one. And this is going to be true throughout the entire process of meiosis 1. All right, so we're on the very first step of meiosis 1, which is prophase 1. The phases are going to follow the same way as mitosis, so we'll go prophase 1, metaphase 1, anaphase 1, telophase 1. And then we'll repeat the same thing, but then we'll have the 2's after it. All right, so we're going to look at prophase 1, and we're going to see what's going on. And if you look at the very first thing is we're going to start noticing these chromosomes. They're going to coil and become visible, just like in mitosis. Just like in mitosis, the nuclear membrane is going to disappear. And again, from mitosis, the spindles will form from the centrioles. And you can kind of see that in the little picture there, is that we can start seeing the nuclear membrane kind of breaking up. That's that little brown little dashed line. You can see both sets of the X's. And you can see that the centrioles and the spindle fibers are starting to form. Homologous chromosomes will pair up. You can see that I have two sets of X's, and they are lining up. The blue and the light purple are one set, and the other set is the orange and the red. And then we get something called crossing over that will occur. And I'm going to talk about crossing over in the next couple slides so you have an idea of what that is. Okay, so we're going to start looking at this pairing and this crossing over, and we're still in prophase 1. And the pairing of the homologous chromosomes is key to understanding meiosis. So you can see that I got two chromosomes. This one I have a blue one and a green one. And they're going to do something called crossing over. And if you notice with crossing over, what you have is two chromosomes. And those two chromosomes will intersect at a certain location. You can kind of see that the blue one is all capital letters and the green one is all lowercase letters. And you can see that big A and little a are kind of facing each other. If you look at the next set of chromosomes, you'll see that they kind of switch some of their information. So you can see that now the blue chromosome has kind of one of the foots to the green chromosome, and the green one has one of the foots to the blue chromosome. And by definition, crossing over is when the chromosomes overlap and exchange portions of their chromatids. And we can kind of see that in the picture. It's pretty obvious there. And we'll continue just a little bit more of why this is important on the next slide. All right, in this side, you can see crossing over one more time, and you can see that it's between a red strand and a blue strand, and they kind of crossed over in the middle. So crossing over can occur in any location, and homologous chromosomes can cross over a multitude of times. It doesn't just have to be once or at the ends. Some chromosomes are more apt to cross over than other chromosomes, so it also depends on the information on the chromosome as well. So what is the point of this? Why is this really important? Why am I covering this right now? The main reason why I'm covering this is because it produces new allele combinations in the offspring. What that means is there's different diversity within the offspring. This is exactly why you might have two biological siblings that don't look much like each other. One sibling might have a different characteristics and the other one might have totally different, even though they were formed from the same two sets of parents. Their appearances are completely different. The other benefit that we have with this crossing over is that it adds genetic diversity. Without crossing over, we would have a lot of people that would look a lot alike each other. Remember, if we kind of trace our roots back down to the same common ancestors, especially here at Turkey Valley, half of you are already related to each other. So you would probably start looking a lot alike. Crossing over kind of makes us look a little bit different from each other. It also does help the human species evolve. So it's kind of also a great thing for an individual standpoint, but also from an evolutionary standpoint. Okay, here we are on my favorite phase in mitosis, or even meiosis metaphase, because it's the easiest one to see what's going on. Remember, that's when they meet in the middle of the chromosomes. And you can see that I have chromosomes in the middle, but it looks a lot different than what we had in mitosis. First of all, you can see that I have two sets of X's lining up instead of one single line, and that's because they're still with the homologous pairs. And those pairs are going to move to the equator. That's kind of the meet in the middle type of thing. So you can see the little picture that I have there. All the black X's are what we call the equator. The centrioles are at the poles. Okay, and the pairs will kind of work together. They're going to try orient one towards each set of centrioles, and so they'll start lining up, but eventually those centrioles and the spindle fibers will attach to them and kind of get them separated. So right now they have a buddy. Eventually they won't. These spindle fibers will also attach to the kinetochore, and we can have some independent assortment, and that's determined on how many chromosome pairs line up among the equator. For us, it's 22 pairs, because then don't forget we have that odd sex chromosome pair that kind of screws things up. Okay, here we are at anaphase 1, and you can see in the picture we have the X's, and they're being ripped apart from their buddy. In fact, they got their arms and legs reaching out, kind of saying, I'm going to miss you, as they go towards each pole. And that's because the spindle fibers will start to shorten. And they're bringing each of one of those pairs to each centriole. So each will go to each end of the cell. And in this case, what's really important here in anaphase 1 versus anaphase 2 is, in this case, the centromeres do not separate. All we're doing is separating buddies. We're not separating at the centromere. And remember, the centromere is that point of attachment of the two chromatids to make a chromosome. So that little dot in that center of the X is not separating. They're being pulled by it, but they're not going to be ripped apart there. All right, we're getting towards the end of meiosis 1, and we got telophase 1. And you can kind of see in the picture that I have down there, you can see a little bit of cytokinesis. In fact, I can kind of see a contractile ring, cleavage furrow. Obviously, we definitely have an animal cell down on the bottom. And what we've lined up is each chromosome at the pole. And what we have is, now we're going to get to the point of a cell that has only one pair or one set of these chromosomes. We're going to start reforming a nuclear membrane around those new chromosomes, and we get rid of that spindle fiber. And you can kind of see in the picture that the nuclear membrane is starting to reform because we got the little dashed lines. I don't see any of the spindle fiber anymore. I just see the centrioles. And in this, we have two chromosomes, each cell, but they're not homologous pairs. This is just one section of them. So following after telophase 1, we have cytokinesis 1. Again, the separation of the organelles in the cytoplasm. We will have the cell membrane elongate. We'll have division of cytoplasm and organelles on each side. You can kind of see the cleavage furrow that is forming there. And you can see that the homologous pairs have been separated at this point. So remember that when we get into this, it's the pinching off the cell to form two. Now, what you're going to get is two cells that are not genetically identical, because those homologous pairs of chromosomes had different pieces of information on each pair. Remember that you received one from mom, one from dad. And now you're going to start splitting those apart. And so maybe one chromosome was from mom, might go on to one cell, and the next chromosome you got from dad went on to the other cell. So these cells are not genetically identical to each other, and that's going to carry on into the next set that we have, which is meiosis 2.